Hi, everybody, and welcome to today's Germination Retail Roundtable webinar. My name is Mark Sinkowitz, and I serve as editor for Germination, and today I'm happy to be your host. Today's theme is making new products faster. We'll talk with four experts who will shed some light on how new technologies are helping and have the potential to bring new products to market for our industry. Before we begin, I'd like to thank our sponsors, 2020 Seed Labs and SeCan, for their support. During the presentation, you'll likely have some questions for our speakers. Please type these into the chat box at any time, and we'll hold a Q&A session at the very end of our webinar after all our presentations are finished. Again, if you have a question at any point, please type them into the chat box. Two of our sessions are pre-recorded today, and one of those people, Ron DePauw, is actually online. He'll be available in the chat. So if you have a question for Ron, please just type it into the chat box. We'll have two live speakers, Logan Skori and Ian Affleck as well, who will be able to take your questions live at the very end of the webinar. Ian and Logan, are you there by any chance? There's Logan and there is Ian. Hi guys, how are you today?
Doing well. Great.
Good. Well, welcome to our webinar. We're looking forward to hearing from you gentlemen very soon. First, we will play our pre-recorded sessions with Kim and Ron. So looking forward to hearing from you guys. So sit tight and we'll chat with you shortly. And just so everybody knows, this presentation is being recorded and will be available for viewing at our website, germination.ca, following the proceedings. Now, we all know that new breeding techniques are helping researchers do a lot of great things like boosting yields to levels previously unknown. For example, according to statistics that were presented at the 2020 USDA Ag Outlook Forum, there's been a 7-fold increase in average corn yields in North America since 1920, thanks to new breeding techniques and discoveries. These are some headlines that I pulled off the internet. So obviously, this is a very major topic right now. Everybody's talking about it, especially in the plant breeding community. But when I was researching topics for this webinar, I wanted to talk with people who could speak to the process of delivering some tangible benefit to the industry and growers beyond simply talking about the potential that these technologies hold. I pulled a federal government document off of the internet, and it plainly says that the process of bringing a new variety to market can still take upwards of 12 years to reach readiness for commercial scale. So even with these new technologies, it can still take a lot of time to actually bring a new product to market. And so I wanted to hear from some people who are working with these technologies and who are really seeing the benefits of this in the real world of being able to bring those new products to market quicker than they might otherwise have. So our first speaker today, who I chatted with just the other day, was Kim Kenward. She is R&D manager for 2020 Seed Labs based in Alberta. She's very involved with the area of seed quality policy and serves as chairperson of the Horizontal Methods for Molecular Biomarker Analysis Committee. In her spare time, she likes to quilt, so she's not all about seed testing. Kim is very multifaceted and is a fascinating person. I really enjoy talking to her, and I hope you enjoy the chat. I talked with her about some new seed tests that have come to market and how these new technologies have allowed her to bring those to market. So enjoy our chat. Thank you, Kim, for joining us on the webinar today.
Well, thanks so much for inviting me, Mark. It's nice doing this as it's a shift from what we normally do, which is a lot of writing back and forth.
That is true. It's nice to be online, seeing you face to face. We're talking today about new technologies, new breeding technologies. Before we get to that, can you tell me a little bit about yourself?
Oh, well, I have a PhD in molecular biology from Queen's University. It actually went through the biochemistry department, which, as any biochemist will tell you, is where molecular biology originated, which is all the DNA testing and interest. So I was actually originally involved in transgenic plants, and I was doing fish antifreeze proteins. So just to dispel that urban myth, we never actually did it in tomato. It was always ever done in tobacco, and it was never released commercially because it really isn't that beneficial in tomatoes. Tomatoes are cold sensitive. They're not freeze resistant, and you need freezing to get fish antifreeze proteins to work. So after I left Queen's, I was at, I did a postdoc at Agriculture Canada, and then I was moved on to doing, I had a lab at Alberta Innovates. Well, it was Alberta Research Council at the time, but now it's Alberta Innovates. And then I moved on to 2020 to commercialize a test I developed. And I've been there since about 2003, I think.
And you are currently the R&D director for 2020 Seed Labs. So you work with a lot of the new technologies that we're talking about here today on our webinar. How have these new technologies, new breeding technologies, etc., made life easier for you and your customers?
Well, it's interesting because when I started at 2020, we actually were the first to build a molecular testing lab, so using the DNA-based techniques to actually detect things, and we maintained that lab. There had been some other startup attempts before us, but they didn't last. We actually came in on the market, we established our molecular lab and just kept moving on. And we built an extensive portfolio of tests using this technology and capability. And so it was interesting because we started from pathogens, trying to detect pathogens. And then as more and more of the new molecular breeding techniques products came onto the market, we built a bigger and bigger repertoire of using the techniques to actually work with the products that were developed through the same or variations on the molecular techniques to create a new thing for farmers and growers to use.
So, Kim, how have these sorts of technologies made things easier for, say, seed growers and farmers specifically?
It's essentially enabling us to do stuff that we couldn't do before and at a much earlier stage. So, for example, our original test was Fusarium graminearum testing, and that had to be determined by plate test because that was the only way to test for it. But the problem with a plate test is that you have to bleach the seed. So you are only really looking at the fungus that's deep within the seed, and that has the ability to grow out. And that occurs at a very early level of infection. With the DNA testing, we could actually detect the fungus if it was present and is starting to be incorporated into the seed at a much later stage of development. So it gave us the ability to detect at an earlier stage than we would have been able to before. And that was really important at the time because we knew that there were parts of the country that didn't have Fusarium infections, and we were trying to slow it down as much as possible in order to let us the chance to get the resistance genetics coming in. And then you've got other situations where you've got like kernel visual distinguishability, which was a great test because you could actually just look at the physical appearance of the seed and tell what class it was. But the breeders were finding that very constricting in terms of what they could do and the genetics they could access to bring into the new varieties. So they didn't want to have to breed in the phenotypic characteristics or the visual characteristics that we were using for determining class. So going in and actually determining variety itself through varietal identification using molecular techniques allows us to determine variety, which means that we can automatically tell class, and they can shift things back and forth between classes much more easily than we could do before.
Now, speaking of DNA technologies, for example, you just developed some new tests for soybean cyst nematode. Can you tell us a little bit about that?
Well, soybean cyst nematode is expected to be a new and emerging threat in Canada. It's been endemic in Ontario. Well, it's been securely in place in Ontario, particularly along, I think, the St. Lawrence for some time now. And then in 2013, we actually lifted the restrictions in terms quarantine restrictions on it. And that was intended to deal with some trade issues, but it also was enabling us to access the earlier maturing soybean varieties, genetics that we wanted. So that's very useful from a seed grower's point of view, but earlier growing means larger acreage, which means a more and looming threat. And if we're bringing the stuff up from the States, where you've got soybean cyst nematode pretty much endemic, then you can expect to get some soil tag that's going to come along, and it's going to come in with a new seed. So you have to anticipate and deal with the threat before it becomes problematic, because there's really nothing you can do about soybean cyst nematode once it's actually established. So the problem with existing soybean cyst nematode tests is it really only looks for juvenile motile forms of the nematode. So you're missing huge amounts of soybean cyst nematode that could be detected if you had the ability to. If you're actually looking for it at a DNA level, that's enabling you to pick up the eggs and the dormant forms, specifically the cysts, which obviously the disease is famous for, and which are just like little sack loads of infectious material once they actually germinate and get moving. So that's a looming threat. It's not one that we could actually address properly using the older test methods, which need a solid amount of nematode in the field for you to be able to detect the motile forms. So going to a molecular technique allows us to expand out and get much more sensitive detection, because we're now looking for the eggs and the cysts and the juveniles. And that gives us more early warning for when the disease might be popping up in the field. And it is popping up, because we have now seen it since the restrictions were lifted, we've seen it in both Quebec at least once or twice, and we're now seeing it in Manitoba, and we have seen confirmation of soybean cyst nematodes in those first identified fields as well. So it's here. It's just here. It could be building. We need to actually be aware of that and take the precautions. So testing is the actual recommendation, that you keep an eye on your on your tissue in the field, and then you test your soils as well.
And so these modern technologies allowed you to bring a test to market that you would not have been able to bring otherwise.
Exactly.
Is there any like science fiction-y type stuff that's really exciting for you? Like when you think about the future of seed testing, like it's like, wow, like maybe one day we might be able to do this far off in the future. Is there anything like that pops to mind that you know, like that today sounds like science fiction, but it's like, well, maybe one day we could actually be able to do something like that?
Have you ever seen Gattaca?
I have not. I've heard of it though.
Well, there's a scene in it where they actually they do a lot of DNA testing on a regular basis to confirm identity and see who's been in places. And she actually takes DNA, the Uma Thurman character takes DNA sample from someone's hair, and she gets it sequenced. So a single hair, it's the follicle at the end, which is the cell that produces the hair. And she takes that to like a little kiosk, and she has it sequenced. And she gets like back inside 10 minutes or something like that, she gets a complete genomic profile and then a list of this guy that she's interested in, his potential good genetic factors and his bad genetic factors. And he actually tests really good on all the fronts, because it's not his DNA, it's fake sample that he carries around with him because he's undercover in these situations. He's hiding because he wants to be an astronaut. But the idea, what they're talking about is next generation sequencing. So we're closing in on that. And the idea is you can just take a sample and you generate a selection of DNA fragments that you've isolated from there, and you sequence all of them And then you run that data through a database so that you can see what's in there and identify what species of creature and living thing is present in there, both plant and bacterial and fungi. And then you can try and figure out what the problem is that's affecting you. This is a really, to me, it's a controversial technology. It's really got a lot of fun aspects of the sci-fi to it, but it's, I think it's a really good discovery tool because it generates huge amounts of data, but it's not a good routine testing tool because it generates huge amounts of data. And it's like, you can't actually tell within that data which is the most important thing that you've found in there that's actually causing the disease, because there's so much other stuff that's in there and it's not strictly qualitative. So to me, it's a good discovery tool, but it's not a good routine analysis tool. And that debate still has to be made to determine whether or not this technology is going to go anywhere. But it's a growing topic within the community. And I've actually come out and sort of said I don't like it for this purpose.
Well, that technology is a little bit far off in the future, so I guess we have a long time to have those debates and those discussions. So thank you so much, Kim, for your time today. It's been great chatting with you, and we'll talk with you again soon. Have a great day.
You too.
And thank you so much, Kim, for that fascinating talk. I have to see Gattaca now. That's next on my agenda of things to watch on television. So thanks again. Now that we've covered off seed testing, I wanted to talk with a plant breeder. And who better than SeCan science advisor, Ron DePauw. Ron is a man who needs no introduction. He's a giant of the cereal breeding community. He was the lead architect of an entire new class of wheat, and he created a slew of landmark new varieties during his long career at AAFC. He developed and registered 57 cultivars of wheat and 6 cultivars of triticale. Over the past 20-some years, 40 to 55% of all the wheat grown in Canada is derived from cultivars that Ron and his team developed. So I was very happy to record a chat with Ron the other day where he really delves into some of these new technologies and how they helped him in his breeding and how they could help breeders in the future. It's about 15 minutes, so sit back and enjoy my chat with Ron, and then we will talk with our 2 live keynote speakers. Well, thank you so much for joining me today, Ron.
It'll be good fun.
So we're exploring this issue of new technologies and time to market and how these new technologies can allow us to bring new products to market faster. We did an article about you in Germination a while back talking about your career, and it was stated in that article that the incremental value of grain produced from your wheat cultivars exceeds $1 billion. So you've been dubbed the $1 billion man of the wheat breeding community. So we knew that you'd be the perfect person to talk to today about this. Ron, thinking back to your early days as a student, what was the first new breeding technology that first inspired you when you think back to that?
I would say it was improved experimental design to predict the genetic value of an individual earlier. So the theory of segregation and frequencies of the desirable genes supported starting selection for both simply inherited traits, which are also called qualitative traits, and complexly inherited traits, which are the quantitative traits, as soon as possible, even in the first generation of selection. And this was new thinking about desirable gene frequencies that was proposed by the late Dr. Len Schabeski, who was the dean at the University of Manitoba. And this was in the 1960s. And the University of Manitoba trained a lot of the plant breeders of that particular era. And the data analysis for this new experimental design used new computers, which were located in the engineering building. It occupied an entire floor of the engineering building. Well, we've got more capacity for computing right now in the iPhone 12. And this was the beginning of the application of advanced statistical analytical techniques to genetic information. This is the path that led to big data and the computerization of plant breeding.
I imagine so much has changed over the years compared to now and back then. And in your experience, how have new breeding technologies evolved over the years? When you think about the breeding that you did and all the new varieties that you created and released, what were some of those evolutions that you recall actually seeing happen in real time?
Well, I would say there's been a multiplicity of changes over time. Like change is really the big constant. And for example, the wheat program at AFC Swift Current from about the late 1970s, we devoted about 10% of our resources to new breeding technologies. And so, for example, the computerization of the breeding program enabled us to handle larger segregating populations with more data. And virtually all of the steps in a breeding program was done without using pencils, pens, or hard copy notebooks. Data was captured using interfaces. Like this is standard stuff now, but at that time, people had a weigh scale. They would put their sample of grain on the weigh scale and then write it down on a piece of paper and then enter it into something for data analysis. Well, with an interface, the weight would be transferred automatically to a digital file that you could then analyze without touching, without having to do anything with a pen or a pencil. And of course, mechanization of plant breeding operations enabled us to handle much larger populations. It's been estimated that we were able to handle about 10 times the number of individuals based on computerization and mechanization. And another major change that occurred was the new NIR technology. And it revolutionized what we could do to measure things like protein content and test weight very, very rapidly. And because we could measure them rapidly, we could select now for grain yield and protein content simultaneously. Because we could, grain yield and protein are negatively related, and we could select simultaneously for the two of them, we developed statistical methods to be able to select and shift that negative relationship of grain yield and protein content. Didn't break it, it just shifted it as a result of better water use efficiency, better nitrogen use efficiency, like building better plants. And some of the varieties that came out of this simultaneous selection for grain yield and protein content were varieties like AC Berry, which won Seed of the Year. Kyle and Strongfield and Navigator Durham, again, Seed of the Year varieties. And recently, AAC Brandon. These are all Seed of the Year varieties, but they are examples of the use of all of these new technologies that came along that enabled us to build superior combinations of genes.
Can you give us an idea of how much more quickly these technologies allowed you to bring some of those varieties to market? Like, can you recall, like, if it wasn't for a certain technology, that variety might have been 5 years, 10 years later getting to market? We might not have been able to bring that variety to market at all. Does anything like that pop to mind?
Absolutely. Like in the case of NIR, being able to select for protein content with NIR, you could do it in seconds. Whereas if you were doing it with the Kjeldahl method, which is a wet chemistry method, it was like an overnight process. to do an analysis. You could only do a handful of analysis. Whereas with NIR technology, you could do, you could do 60 or more samples in an hour. So it was just really revolutionary. But I think we have to be very much aware that wheat is a self-pollinating species. And a new variety has to be homogeneous and homozygous. So it's breeding true. This is achieved by inbreeding. So you have to go through the generations. It's like a pregnancy. No matter what we do with all of our new medical interventions, a pregnancy normally is still 9 months. The same thing with wheat. You have to make it pure breeding, to make it homozygous and homogeneous, you have to go through these generations. But having said that, there is this, what we call the acceleration of generation advancement becomes key to reducing the time to market, acceleration of the generation of advancement. So multiple generations per year can be grown by using contra-season nurseries, greenhouses, growth rooms. And we've been, one of the things that we developed was to be able to take a seed that had, flowered, and then within about 15, 20 days, you could break the dormancy and start it over again. And we call that acceleration of generation advancement. But the buzzword today is called speed breeding, but it is essentially the same thing.
Wow. So when you think about the future, like science fiction type stuff, what do you see coming down the pipe? Like, I've heard all kinds of things that, well, we can bring new varieties to market faster. Yes, we can, of course, but how much faster? Like, breeding is breeding is all about time, of course, as you know, but could we one day bring a variety to market in like a year or 2 years? Or like what do you think might happen, when you look down the road?
Well, both the common wheat genome and the durum wheat genome have been sequenced. And there's a lot of work going on to understand, what is the functional part of all of this DNA? So there's a lot of work going on in that area. And that's going to be fundamental to what I'm going to get to. Another big thing that's going on is that the cost of genotyping has really come down, making it possible to have a lot more genomic information. And then we have continued development of computing power. And we've got this high throughput phenotyping. They're developing ways of using the drones and the imaging to get these spectral, to get it to be three-dimensional, to be able to get the information. And because of the knowing more about the sequence and the functional purposes, that becomes foundational to be able to do gene editing. There's all of this stuff about gene editing, but unless you've got a clone gene and you know what it functions and you know what the pathway is, you won't be able to really do gene editing, in a practical way. So I think we're having the possibility of moving in genetic enhancement to level 4.0. That is breeding by design. That is, we're going to have the ability to combine any known alleles into an optimum combination. However, all of these are merely tools to assist in the prediction of the superior gene combination. Multi-location testing will still be required. Proof of performance of a superior gene combination can only be done in real-world fields of exposure to all of the stresses of heat, drought, could be a cold seed bed, could be a wet, soggy soil. It could be cool fall weather conditions. That's all got to be proofed out in the real world. So grain in the bin that can be processed to make nutritious food, products at a profitable price to producers is going to still be foundational. Everything else, I would say, is predictive.
We've learned a lot coming out of the pandemic. It's fascinating to me how all these areas are interrelated. Before I let you go, Ron, one last thing. Why are all these new technologies so important for SeCan specifically?
Well, the expectation is that the new technologies will enable the assembly of superior gene combinations faster and more accurately. They'll be like, yeah, an example of gene editing that I'm going to give you. So it is possible to have a very robust variety. And let's say it's a midge-tolerant variety. I'm going to use that one as an example because we know there's only one gene that confers resistance to the orange blossom wheat midge, called the SM1 gene. So, and we know mutation is real in all living organisms. So if the orange wheat blossom midge was to develop a mutation and the midge didn't catch it and it reproduced, and now we've got a population of midge that are developing virulence on SM1. Well, the SM1 gene having been cloned, they could look at what is it about this mutant midge that is able to overcome, so that you're going to have some metabolics that are going to be going on in here. And then they might be able to say, okay, if we change this base pair or that base pair, this mutant midge will no longer be able to overcome the SM1 gene. But in the meantime, this now becomes a gene-edited SM1. So it's like a SM1AB, something different. So that's what would be an example of how to use gene editing to make a new variety. And so in terms of recent new products that have come on using some of this that has been possible are the SM1 high yielding, SM1 tolerant or midge tolerant wheat varieties that are high yielding, very good disease resistant. The robust products are the AAC Starbuck and AAC Wheatland are examples of this. And of course, these varieties wouldn't have been possible without the producer checkoff, which has been in place since 1995. The farmers have been investing long-term on this. It's enabled all of these new technologies to be implemented. And there's been a huge return on investment as a result of that. But in addition, of the producer checkoff, there's been other funding agencies like the Sask Ag and Sask Ag Development Fund and ACID-F. And ACID-F was involved in co-funding the development of the AAC Starbuck and AAC Wheatland. So funding will continue to be super important.
Fascinating stuff. Thank you so much for your time today, Ron.
You're welcome, Mark. You're welcome.
Yes, thank you so much for that, Ron. Fascinating stuff. And again, Ron, I believe, is available in the chat box. So if you have a question for Ron, please just type it in. You can also send him an email at rdepauw, RDEPAUW @secan.com. We are just over halfway through now. We're just going to switch over to our two live keynote speakers of the day. I wanted to talk with someone involved in genomics, and I decided to reach out to Logan Skori. He is CEO of AgGene based in Alberta. He has expertise in the field of gene editing and development of genetically engineered crops. Welcome, Logan.
Hi, how are you doing?
Good. How are you today?
I'm doing pretty good.
Excellent. Well, thanks for your time today. I believe you're going to share your screen with us. You're going to talk a little bit about some of the exciting work you do, a little bit about AgGene and some technological developments in the field of gene editing, etc. So the floor is yours, sir.
Thank you.
Yeah, you might have to share the specific window.
There, how's that?
Looks good. You might just have to, maximize your
Maximize?
screen and then perfect. There we go.
Perfect. All right. So first of all, thanks to Mark for the opportunity to be speaking today. My name is Logan Skori. I'm the CEO of AgGene, and I really want to share our story and some of the exciting things that we're working on in the gene editing space. Before I get started, I'd really like to introduce the team that we've assembled. So these are the co-founders of AgGene. So I had the opportunity of growing up on a grain farm near Kinsella, Alberta, and I'm currently finishing a PhD at University of Calgary with a large focus on plant biotech. During my upbringing, I spent a lot of time riding around on the combine, and a lot of times I was disappointed with the way that crops performed, oftentimes due to abiotic stress, and I think it really motivated me to pursue an education and be able to help develop technologies that will help people like my dad and their crops. The next person that we have on our team is Dr. Marcus Samuel. He's a professor at University of Calgary and my PhD supervisor, and he provides a lot of great insight on the tech developments that we're currently ongoing. And he's been in the plant biotech research field for a couple decades, so he's always got lots of great insight. Next, we have Rick Rutherford. He's a grain grower, a seed grower based out of Grassy Isle, Manitoba. Rick brings a lot of expertise from the seed business, so he's always great to chat to. And last and certainly not least, we have Sarah Foster, who's the president of 2020 Seed Labs. Sarah, I'm sure everybody in the Canadian seed industry knows Sarah, and she's always got lots of great insight and business advice. So one of the major problems that our company is focused on is addressing the need for plant-based protein. So there are a number of different factors right now that are really driving the demand for plant-based protein. Amongst these would be climate change, the reduction of arable land, increasing population, and quite a recent fad is people kind of transitioning their diet to a more plant-centric focus. So the combination of these factors is really boosting the demand for protein, and by the year 2050, I think the demand for protein is supposed to double. So we have to start thinking of solutions to maximize protein production on the same amount of acres that we currently have. And so to do that, we're actually leveraging gene editing. So during my PhD, we developed a proprietary genetic technology. So we recently filed a provisional patent on this pathway, and it really encompasses the use of gene editing to develop a trait which would ultimately be integrated into breeding programs and with the desired outcome of high protein crop cultivars. And just to give you a little brief background on the technology. So it's important to know that seeds accumulate a lot of different proteins during the later stages of seed development. And these are really important for a number of reasons, primarily for nutrition when that seed is dispersed into the environment and it needs to germinate. But there's also protective functions of various proteins. And so we've done a lot of work in this particular field, and we've really focused our attention on a gene, and I'll call this gene A. And what we find is the protein accumulation is partially regulated by gene A. And when we have increased activity of gene A, we're going to see an increase in the protein content of seeds. And so with some of our previous research, we had developed transgenic canola varieties. And what we found when we looked at the protein profile, we noticed a 20 to 25% increase in the crude protein content when compared to the untransformed variety. So this was very exciting for us because this is obviously a substantial boost in seed protein. But the one drawback of this was it is a GMO technology, and given the regulatory landscape across a number of jurisdictions, it's not really a favorable technology to pursue. And so we had to look at alternatives, and we started to ask the question, how can we utilize gene editing to help us develop a technology and help us develop this high protein trait? And so we did a number of different things, obviously extensive investigations into the literature, bioinformatic analyses, and we found a handful of candidate genes which actually kind of inhibit or restrict the function of our desired gene A. And so we started to really strategize on how we can increase protein. And so we're actually utilizing gene editing to remove the function of gene B and gene C, which have a negative effect on gene A. And when we can actually remove these particular targets, we should be able to increase the activity or function of gene A and therefore increase the seed protein content. Now, interestingly, we've done a lot of preliminary studies where we investigate the mutant varieties of gene B and gene C in Arabidopsis. And when these genes are disabled, we're seeing protein increases. For gene B, we see about a 5% increase in the protein content, and gene C, we see about a 10% increase in protein content. So this is obviously very optimistic, and we have, a few other avenues that we're also exploring. The one value of this technology is this pathway is a fairly conserved set of genes across monocot and dicot plants. So, we're looking at expanding the technology into soybean and cereal crops, including rice. So that's obviously a very interesting development. And just to touch briefly on gene editing, I mean, this is the part that is really exciting. And right now in the commercial setting, there are a number of different gene editing systems that companies can use. Obviously, the most predominant and most popular would be the CRISPR-Cas9 system. And obviously we've got our own ideologies on how we want to do gene editing, and we've chosen to pursue a gene editing system called MAD7. And this is something that I'll talk about in a minute, but it's important to note that gene editing systems have two really important components. The first component is called the guide RNA. Now, this is depicted in pink in the cartoon. And this guide RNA is a customizable fragment of RNA. And basically, we can design this guide RNA to target basically any gene within a genome. And this guide, once it is in the plant, its job is to find the area that we specifically want to edit. So this kind of directs the events of gene editing. And the second major component is the nuclease. And so the nuclease is going to interact with this guide RNA on a scaffold, and the nuclease is actually going to cut the DNA. So the nuclease is like a pair of scissors, essentially. And as you can see in the cartoon, there's two strands of DNA that will be cut during the gene editing process. And when both of these strands of DNA are cut, the plant or the organism that is being edited is going to go into repair mode. And because this repair process is not 100% efficient, we can actually gain or lose small pieces of DNA in the process. And when we start altering the genetic sequence of a gene, now we start to alter function. So the most likely outcome of gene editing is you're actually disabling the function of a gene. So ideally, you would want to disable the function of genes that have a negative impact on plant development. And to briefly touch on the system that we're using, so we are currently developing the MAD7 gene editing system. And this was a technology patented by Inscripta Life Science, and they have actually provided the use of MAD7 with no royalties and no licensing fees. And for a small startup company with limited resources, this is a very powerful technology, simply because we don't have to go and license Cas9 from Corteva. So we have a nice freedom to operate. But again, this is a long journey to develop a gene editing system. And I'm really happy to report that we have built the system and we have successfully tested the MAD7 system in vitro. And all this means is we purified the components. So we have the guide RNA and the nuclease, and we incubate it with plant DNA in a test tube, and we were able to see gene editing occur. And so right now we do have a canola variety harboring our MAD7 gene editing system. So we're really excited to characterize, and hopefully within the next few months, we'll be able to confirm that this is a viable system for our development. And just to kind of wrap things up, I'd like to just briefly talk about the advantages of gene-edited crops. To me, one of the most important advantages is this is a very precision-based technology. When we look at other plant breeding techniques such as mutagenesis, we have no control where changes are being induced in a DNA blueprint. But with gene editing, we can be very targeted with where we want to make changes. And depending on the system, there's very little chance of off-target effects, which I think is also important to consider. Due to the precise nature of this technology, it also allows companies to rapidly develop. So you have to spend less time cleaning up a genetic background. And this is obviously important, as it will reduce operating costs and some of the long-term costs associated with plant breeding. And with the good always comes some of the challenges that companies face bringing these gene-edited technologies to the marketplace. The first would be the regulatory concerns. So right now, there are a number of different jurisdictions, and every jurisdiction has its own view on gene-edited crops. This is something that Ian will discuss more in the next talk, but it is certainly something to be mindful of. The second would be the intellectual property. This is a very hot area of research, and there are a number of patents being filed on a daily basis regarding gene editing. So, as a company, you have to be very mindful of other people's IP, but you also have to do your part and protect the IP that you develop as well. And last, and certainly not least, the market acceptance is everything. We need to have a market and a destination for these products, and people need to be accepting of the technology. And I think the large companies that are really focused on gene editing have done a really nice job telling the narrative and the power behind gene editing and how we can develop traits that ultimately are going to enrich people's lives, especially when we start looking at things like added nutrition and food security. So with that, I'd like to thank Mark for the opportunity, and thanks to the other speakers for a really engaging event. And if people have questions offline, you can certainly feel free to give me an email. Thank you.
Thank you very much, Logan. If you have a question for Logan, please just type it into the chat box. We will take a couple of questions at the end. Before we take questions and wrap up for the day, we are just going to hear from our final speaker of the day. Last but not least is Ian Affleck. Ian is Vice President, Plant Biotechnology for CropLife Canada. Welcome, Ian.
Thanks, Mark, and thank you very much for the invite. I'm just sharing my screen here now.
Yeah,
Let
excellent.
me know if that's showing up all right.
I can see it fine. And Ian, you're going to talk a little bit about something Logan touched on at the very end of his talk, regulatory issues, all of these fascinating new technologies that we've been discussing. Really, it's hard for them to really go anywhere or be of any use if we don't have a regulatory environment, of course, that can allow people to use these new technologies to actually bring products to market. So take it away, Ian.
Great, thank you. Yeah, I'll focus on kind of the regulatory landscape as well as the public engagement and transparency side, and give an overview of what's happening in gene editing in that space. So just move to the next slide. So as mentioned, I work for CropLife Canada. We represent small, medium, and large sized companies, both from Canada and globally, that work in the plant science space, both developing, manufacturing, and distributing plant science innovations. So typically when I give these presentations, I'm not usually set up by actual experienced plant breeders and scientists. So I'm kind of trying to do some of that storytelling. So I can skip over that as it's been very well done. But I'll mention a few things just context, because the regulatory and the engagement approach does it leads out of how we understand plant breeding over time. So usually I'll talk about these kind of examples of how wild emmer is kind of an ancestor of wheat and how we got it to common wheat, how we took teosinte to corn, how we took banana from something that was largely inedible to something that is mostly fleshy edible elements, as well as how genetic selection allowed us to make all of these different products from the same thing, from wild mustard, Brassica oleracea, if you will. And I think kind of linking back to what Ron was saying, part of the reason to tell this story as we head toward the regulatory and the engagement space is understanding that often gene editing becomes the star of the discussion when the plant breeder and the scientists who are utilizing that tool are really the star of the discussion. And a quote that I heard once is, when someone builds a beautiful home, we don't celebrate the hammer, we celebrate the plant breeder. And I think gene editing in this space really is the hammer of the example, and it's the plant breeder that's applying it in order to get the outcome they're looking for. And I thought Ron mentioned it as well in detail, that if you don't understand the gene, you haven't located it, you haven't cloned it, you can't utilize it, then gene editing really isn't going to be much use to you. So another analogy I heard is, gene editing is not a magic wand. You can't just hand it to anyone and expect it to have magic and suddenly you have a variety. It needs to be in the hands of a very experienced plant breeder in order for it to be useful. So it's much more, going back to the hammer analogy, it's much more like a really fancy laser guided drill to help them do the work that the plant breeder was doing anyway. But in the context of gene editing, there has been a number of recent papers and science, which I think tells the story really well. One I would like to highlight, this is if you'd like to go back and read into this much more, recent paper from Agriculture and Agri-Food Canada called Genetic Variation and Unintended Risks in the Context of Old and New Breeding Techniques, really describes the history of plant breeding, how this all builds on itself, and how a plant breeder is going to incorporate these new techniques into what they already know. Another great paper coming out of the U of S, Advanced Domestication, Harnessing the Precision of Gene Editing and Crop Breeding, another great paper that describes how this can be so useful in plant breeding. And one item that I pull out of that paper, they had one really great graphic here as you're going through all your F hybrids, looking to get that great variety. And in this space right here specifically, you get to your advanced field trials, and then you're gone from thousands of crosses down to 100. 80 of them just don't meet the quality standards. And this is where the paper describes how gene editing could save these near misses. You could go in and fix the little thing that didn't make it and pull it back onto the shelf, and how much efficiency that could add into a breeding process would be quite impressive. So this isn't the really headline grabbing high fiber wheat or the really cool, high oleic oil soybeans. This is just helping breeders become more and more efficient in what they're doing and losing less and less of their crosses to, as was described in the paper, near misses. We're kind of coming off of that. So understanding that gene editing is building on a long history of plant breeding, what is our regulatory landscape globally, and where is it going? So before we can talk about regulation, we need to understand the status of safety. It is largely accepted that gene editing of plants is as safe as traditional breeding when you're going for traditional breeding outcomes. One of the first really great coverage of this was EU Federation of Academies of Sciences and Humanities. Basically, all of their universities, all of their research spaces published a great coverage of how we can be confident that gene editing, when used along with traditional breeding or for the same outcomes, is safe. That's been followed up since with a number of reports from a number of government and academic institutions, even one here in the bottom right, Genome Editing for Food Security and Environmental Sustainability out of McGill University in Canada. just further demonstrating how this builds on what we know about plant breeding. So now that we have that basis and regulatory bodies are understanding the long history of safety in plant breeding driven by the plant breeder, how does this tool fit in? We've seen Australia, New Zealand, Japan, the US, a number of South American countries have all come forward with a similar regulatory approach of saying, leaning on our familiarity with conventional plant breeding, provided you're not adding foreign DNA to the product, we can leverage our long history of experience with conventional plant breeding. If you did add foreign DNA, we're not saying that's inherently risky. It's just that there's less familiarity with what traits that can produce. And therefore, there's different levels of regulatory oversight that's needed. So it's not about one is safe and one is unsafe. It's about saying that we have familiarity with one and less so with the other. We're seeing that kind of general regulatory trend take place in most countries around the world. Now, there's two major regions I didn't mention. One is the EU and the other is China. The EU is locked in probably a 2 to 3 more year political and regulatory challenge to update their regulations. They have published a paper saying they are not fit for purpose. The existing GMO framework should not be applied to gene editing, while it in some ways does today, but hopefully will not in the future. But they're going to be a number of years before they get that figured out. China will likely require some level of oversight, but how much oversight that is, whether it's notification or whether it's actually for submission, that's yet to be seen. But when looking at the cost of introducing a product to a marketplace around the world, most countries are looking for notification of the product at most. So hopefully this will keep the cost of introduction at a reasonable rate so that we do have a broad variety of companies that are able to work with this technology and bring new products to market. In the Canadian specific regulatory environment, Health Canada has already completed a public consultation on updated policies. We're awaiting now the final publication of that, which will probably happen after the election. Important element of that is there's a voluntary transparency initiative to facilitate companies posting prior to commercialization new gene-edited varieties as a tool for public communication. And we'll talk about that a little farther in the next slides. Canadian Food Inspection Agency also working very hard to update their policies. The PNT, or environmental release element, is currently under public consultation. I would recommend anyone who works in the plant breeding space check out the CFIA website, read those policies, as remembering the Canadian policies are not strictly for gene editing, they're for all plant breeding, because in Canada we're product-based, not process-based, so all plant breeding falls under these regulations. So I would recommend you read that to see what you think the impact of these proposals could be for you. Health Canada's are quite good, very focused. I think it has it's a good refocusing of the Canadian approach, and currently out with consultation on the environment side. Novel feeds consultation expected in the fall. But overall, Canada heading in the right direction. Hopefully, we'll see all of these policies be finalized in early 2022. But just one example out of the Health Canada review, very strong statement of safety from their literature review that the use of gene editing technologies does not present any unique safety concerns compared to other methods of plant breeding. So very positive there. So finally, just finishing off, as you see regulatory systems evolving and new policies coming forward, what comes with that and with new technology in general is public engagement and transparency. So on that, CropLife and other seed industry representatives did some research into how do we engage with the public, and we learned a couple of key things. 61% of Canadians were completely unfamiliar with gene editing in any context, whether it's human health, plant health, anything else. This means there's an opportunity to engage early and provide good information. So now is the time for that active communication, recognizing it's a very competitive landscape for audience time. We're not just competing on science here, we're competing with TikTok and YouTube and people falling down on the internet and how that gathers people's time. If you want them to take time to learn about plant breeding, how are we going to grab their attention? And we need to grab their attention because first contact with accurate information is going to be critical to these products getting market acceptance. So to do that, modern communication practices are required. In today's day and age on the internet, you have to entertain first and foremost, and then you have the opportunity to inform. So grab their attention with benefits and potential, then tell the story. What is plant breeding? What's modern plant breeding? And then gene editing. We saw so many in our research that didn't know plant breeding even existed. Then you can tell the science, the safety, and have always targeted for your audience. So just finishing off, taking all that into account, CropLife Canada and Seeds Canada, along with a wide variety of ag stakeholders, launched a communication initiative called Nature Nurtured, Real Food Innovation Through Gene Editing, supported by over 29 different ag associations, from grain elevators to grower groups to life science companies to the CFA, right across the board, demonstrating that there's value in gene editing and we don't want Canada to miss out. And this really helps provide legitimacy to communicating with the public. You'll see this kind of stuff on social media, fun, punny advertisements to kind of attract people into the website, wake up and smell the coffee plants, really funny dad jokes like "I'm soy into you" and little stories about what we're doing with gene editing. Animations like this that kind of are fun and inviting, make people want to learn about what we're doing in the plant breeding industry. Hopefully they'll click on these when they see them in social media. It takes them to the Nature Nurtured website where they can learn about what it is, why it's beneficial, why it's safe, some stories, FAQs, and learn who is supportive of the technology. And finally, you can follow that social media campaign and share if you're available at naturenurtured.ca, Nature Nurtured on LinkedIn, and the same on Twitter. So I went through that very quickly, Mark, to try to hit your 2 o'clock time frame. But overall, we have bumps in the road for the EU and China potentially, but not as bad, we're hoping, as we saw with GMOs. And overall, the industry is trying to do a better job of getting out there early and communicating on gene editing, learning from the challenges we had with GMOs in the past and waiting too long to engage.
Thank you so much, Ian. That was terrific. We have a couple of minutes left. We can take some questions from the audience. If you have a question for Logan or Ian, please type it into the chat box. Logan, we had a question regarding the difference between Cas9 and MAD7. Can you enlighten us a little bit about the exact difference between the two, if possible?
Yeah, that's a great question. So MAD7 is actually closest to another gene editing system called CPF1. This is another variant of like a CRISPR-like gene editing molecule. The major difference would be it's a different sequence. So when we look at the amino acid sequence that composes this, it would be drastically different. But when we actually look at the function, one of the major differences is they have different recognition sequences. So there's a small, there's like a 3 or 4 base pair sequence that has to be close to where you want to edit. And for Cas9 and CPF1 and MAD7, those are slightly different. So that makes a big impact on where you can actually do gene editing at.
I have a question. I'll direct this to you, Ian. Do some crops have higher funders, do some crops have funders who are more accepting of GE technology, for example, canola, versus other crops like wheat, for example?
I think crops who have already had experience with GM technology are probably already more familiar with, these kind of breeding techniques being integrated into their crop space. But I don't think when you look at other crops like wheat, barley, oats, others who didn't get to benefit from GM quite as much, I think it's really the value proposition of the trait that you can bring to the market. Is it needed? Is there value? Is there value to the grower or the processing chain or to the consumer? And I think if the value of the trait is there, then the effort required to ensure that there is a market for it to go into will follow. But if the value of the trait is not high enough, then you got to balance that with the effort that commodity chain is going to have to put into ensuring its buyers and sellers are ready for that product. And then, of course, export markets are going to matter. If you're 100% exported to the EU and things are still a mess there, then that's going to make things a little tougher. But hopefully, with the rest of the world, hopefully the tide is pushing in the same direction, with the rest of the world all aligning with similar approaches, like we're seeing with Health Canada and others, that we can have some positive influence on the outcome of this regulatory review in the EU, for example.
Yeah, in terms of the regulatory review, Logan, is there some what do you need for a technology like yours to be able to bring new products to market faster? And in terms of our regulatory system here in Canada, what are you looking for in that regard?
Well, there's certainly no doubt that safety should be a priority. with any jurisdiction. I think that's very important. But I think being recognized as a safer alternative to GMO crops, being subjected to the same regulatory aspect in Canada is potentially frustrating, especially in the U.S., where I'm not sure if it's the USDA or the EPA, but they do have kind of a more streamlined regulatory process. So I think it's just, setting the bar and being consistent.
Yeah, it's when you look at this patchwork of different approaches around the world, Ian, with regards to the European Union, that's made the EU's made a lot of headlines in recent months after it decided that gene-edited products would be treated the same as GMOs. Do you see any potential progress there in the future? Possibly, maybe they might do things differently. What are your thoughts there?
It's going to take time. They're in quite a mess because it was a legal body who said the legal interpretation of the regs is this, that gene editing is a GMO, because their exemption for conventional breeding did not say the words gene editing, although you could argue that this is a form of mutagenesis, so it should be included. But even in that full legal opinion, if you read it, one of my favorite parts is the judge says, we understand that this doesn't make any sense scientifically. I'm paraphrasing. But we're not scientists, we're legal authorities, and this is what the rule says. Whether it makes sense or not is not our problem. But that's where the science body just came out a couple of months ago and said, yes, this reg framework is not fit for purpose if it must be applied to gene editing. So we have to do something. The Council of Ag Ministers said, yes, let's do something. Now, what are they going to do? That's where we hope we have an opportunity for it to be better. And there's a lot of different examples they can take from around the world, whether it's the Health Canada approach to voluntary transparency, whether it's Japan's approach to a voluntary but very expected notification process that's not a review. But a lot of opportunities for other countries to demonstrate to Europe a very functional way to do this. But it's going to take them about 2 years at a minimum to get this straightened out. And in the meantime, it's going to be a little bit of a gray area of what the EU would or wouldn't do with a gene-edited product should it come to shore.
Yeah, well, and that's where efforts like Nature Nurtured are so important. Thank you, Ian, for putting the social media info up there in the chat box. It's efforts like Nature Nurtured that will help to educate the public, and because as we all know, it's those younger people who will eventually become those legal experts that you referred to, and so that's really the way we can help change our system is to educate the public and get them to realize the scientific truth of these technologies and how they work. I have a comment here that the outreach efforts seem to be mirroring what we saw with GE. What confidence does the industry have that engagement will be effective as opposed to polarizing? I'll open this up to both of you before we sign off for the day. How do you see that debate shaping up?
Yeah, I think I could say I think the difference here is we've learned a lot from what was polarizing in the attempts on GM. I think we went really science heavy. The biggest thing that I've learned from this is that I, as a scientist, need to listen to the communication professionals and be like, no, but I have this paper. And like, well, nobody cares. That's not what they're interested in. They want to know that their coffee is going to be impacted and there's a reason why they should care. Once they care, then you can tell them about the science down the road. But you've got to connect on some values first. You've got to connect on something they're interested in. And that was what's really hard. In some of the campaigns, we're talking about bananas, the Cavendish banana. We're talking about coffee. We're talking about chocolate. People are saying, well, those aren't really Canadian crops. And it's kind of, well, people don't visualize a handful of canola. They visualize the coffee they drink every morning. Now, once you have them in, then you can educate them on the importance of canola, the importance of wheat to the Canadian economy. And then here's how it's going to help that. And for climate change and helping farmers manage climate impact. But you've really got to entertain them first. And I think we missed that early on, where our information was sometimes a little dry and factual. And the stuff that was fear-based was pretty entertaining. And we got beaten to the punch. So if we want to get out there with the accurate information, we have to learn how to be more entertaining. So that's something I've taken from the comms professionals that we've worked with in launching the campaign. Hence the the groaner dad jokes that we use to try to be approachable.
Yeah, it's like that old media saying that if it bleeds, it leads. Unfortunately, sometimes the scary information can be the most entertaining, and so that's part of the challenge is to try and cut through that and to get people to pay attention, even though something might not be as entertaining or as dramatic or as colorful as some of that misinformation that's out there. Once you're able to differentiate between the good information and the not so good information, that can be half the battle is to teach those media literacy skills. And so it's efforts like Nature Nurture that are so important to that. So thank you, Ian, and thank you, Logan. I think that about wraps up our time for the day. So unless either of you gentlemen wanted to add anything else, I will wind things down here and just say thank you so much to our audience for joining in today. Appreciate you spending your lunch hour with us. Again, I just wanted to thank our sponsors, 2020 Seed Labs and SeCan, for their support. This webinar will be available at germination.ca in the coming hours. Ian and Logan, thank you so much for your time today.
Thank you.
Thank you.
Have a great day, guys, and have a great day to everybody out there watching right now. This is Mark Zinkowitz of Germination signing off.